But I wanted to go through this again and just kind of refresh your memory about what we were talking about yesterday. The majority of our energy in this whole process is going to be, is it like an accordion in the hall? Or?
It helps you do this.
All right, I'm back. The majority of our energy is going to be obtained in the third part of our stage of cell respiration, and that is our oxidative phosphorylation that happens in that inner membrane of the mitochondria, or the cristae, and it's happening through a series of molecules, proteins, that are embedded in the inner membrane. It still seems really loud. I don't know. It's next door. I don't know what they're doing. Those NADHs, as well as the FADH2 that we had in the Krebs or citric acid cycle, are carrying electrons. And those electrons, you can think about those electrons as they are energy. If you have a ball on a string and you start twirling it around, kind of like a lasso, there's energy associated with that. If you swing that at somebody and you hit them in the head, it hurts. Right? There's kinetic energy. There's force behind that. And that's what's happening with the electron. The electron is spinning and it's revolving around the core of the atom, whatever molecule it's around, and it's being passed, that energy, that electron is being passed from one carrier to another in those series of reactions we call redox reactions, where one picks it up, becomes reduced, and the other one gives it off and it becomes oxidized. The goal of that is to get those electrons to this point in the mitochondria where they are passed from one carrier to another. And as they do so, they're going to pump out, they're going to give up a little bit of energy as they move from one carrier to the next. And they use that energy to pump a hydrogen ion against this concentration gradient. It's just me, or is that loud? It's loud, right? It's so heroic too. I want to stop and do something.
Talking about the electrons, I'm doing the whole thing.
What's going on? It happened last period too, but it's a different teacher, so I don't even know what's going on. Yeah.
What is the Q?
The Q is our unlabeled electron carrier. So it is, that's a great question. Q is becoming reduced very briefly because NADH is giving up its electrons, giving it 2Q. And in passing it 2Q, that transfer releases a tiny little bit of energy that can be used to pump, to push that proton, that hydrogen ion against its concentration gradient. Okay? You don't have to remember Q. Q has a name. We're just going to call him Q. That happens once. And that electron's going to get passed again because Q's like, nope, hot potato, I don't really want it. It's going to pass it on to another carrier, which is called cytochrome C, or site C here. Again, you don't need to know those names. It's just another carrier, just a little intermediary. And that pumps another proton. Little bit of energy gets released. If you swing a ball on a tether and you only swing it once, what happens? It eventually is going to lose its energy, right? It's going to go a couple times maybe, and then it's going to boop, it's going to kind of lose it. So there's a one final electron acceptor at the end, and that final electron acceptor is going to be oxygen. And it's going to take that electron when the electron is pretty much completely de-energized. And it still is an electron, it still has a little bit of energy in it, but not enough to be really useful to us. So the electron will get picked up by the oxygen along with a couple of protons, and it will become water. I forgot to say that when that happens again, we pump another proton through. Okay. So this creates a situation where we have a whole bunch of H pluses or protons on one side of the membrane, and they would very much like to go back into or across the membrane, but because these are ions, they are not easily diffusible through the phospholipid membrane that we have here. So they can't. And the only place that they can do that is by going through this thing called ATP synthase. And so all my H pluses will kind of queue up here. And they will go back into the cell. Not cell, sorry. They will go back inside the membrane and down their concentration gradient through ATP synthase, which is an enzyme. It spins. And as it spins, that spinning creates kinetic energy that is used to produce our ATP. So we got ATP here. And what I was going to show you is the idea that it's kind of like a wagon, a water wheel. So that's just a water wheel, and the water is running over it. And as the water runs over it, if I'm using this as an analogy, what is the water from chemiosmosis? The hydrogen. Yeah. So the hydrogen is flowing through the ATP synthase. That's the water wheel. And as it does that, it will generate ATP. In this case, it's actually being used to generate electricity, which I think was pretty cool. Yeah, I think it's electricity. Anyway, he's got a little generator there. And so this is an old-fashioned, sort of old school way to generate electricity. But we still do this. We still have hydroelectric dams or hydroelectric power. So some of our electricity comes from this, from the water pushing the wheel and generating that. So it's the same thing that's happening in our ATP synthase. It's turning, and as it turns, it phosphorylates ADP, turning it into ATP. Okay. Questions on that? I think I have another picture of that. As the hydrogen ions go down their concentration gradient, that is what we call chemiosmosis. Okay, the physical movement of those hydrogen ions is called chemiosmosis. There are other examples of chemiosmosis, not just this one, but this is one example of it. There's my my wagon wheel again, and this guy spins. And as it spins, it causes this to spin in a different direction and produce my ATP. So it phosphorylates ADP. The movement of the hydrogen ions is called chemiosmosis. The production of the ATP through this process is called oxidative phosphorylation. We're phosphorylating, just like we did before, the ADP to become ATP. But now we call it oxidative because, in part, because of the fact that our final electron acceptor to get all this done is oxygen. So it's using oxygen to create all this, to do all this. Let's see what that says. Certain electron carriers in the electron transport chain accept and release hydrogens along with electrons. Those are the, like Francis said, the whole Q. So Q is actually ubiquinone. You don't need to know that, but that's the electron carrier that they're talking about. And cytochrome C, or the CYT-C that we saw, that's another electron carrier. In this way, the energy stored in the H positive gradient across the membrane couples the redox reactions to the electron transport chain and that to ATP synthesis. Final electron acceptor is oxygen. What would happen if there was no oxygen available? Think about the...
Electron reception.
You wouldn't be able to make ATP? Because why?
Electron because you recepted.
So where are they going to back up? What's not going to be able... Yeah, so glucose is going to happen, right? Because glucose does not require oxygen. But after that, all those NADHs, they're going to be full, right? They've got their electrons. Like a taxi. A taxi's busy. You don't pile more people into the taxi. It's just busy. You have to wait, right? So if there's no oxygen there, this whole system's going to back up because the NADHs cannot become oxidized. They cannot give up their electrons because there's nothing to give them up to. There's no oxygen. And that is the real story of why we need oxygen, for everything. If we go too hard, we can't generate enough oxygen to generate enough ATP. And if there's not enough oxygen in our lungs, then we cannot generate the ATP. And it's kind of like a catch-22. Right? So you have to slow down when that happens. Let's say you're running, as fast as you can. Eventually, you're not going to have enough oxygen. If you don't have enough oxygen, you can't dump off those electrons and you can't generate more ATP. So long story short, you have to slow down. You slow down, you breathe more normally, you are using less energy now, you're needing less energy. More oxygen is flowing into your lungs, and now the electrons are able to be dumped onto the oxygen to become water. Okay? The other thing that I think is cool about this whole thing is the oxygen that you breathe that you breathe in becomes the water that you sweat out.
Part of the H2O.
That's going to be used. That is also going to be partly sweated out. It's also going to be used for hydrolysis. So you've got all these chemical reactions, right? Because all these things require water to break them down. Yeah. And cray. Yeah. All right. All right. Okay. During cell respiration, most of the energy flows in the sequence from glucose. Most of it, right? We get a little bit of ATP through glycolysis and electron transport, glycolysis and oxidation of pyruvate and citric acid cycle. But those three events don't get most of our ATP. Most of it is going to flow from glucose to NADH and then to the electron transport chain. And then that word here they're saying proton motive. They're talking about the water wheel. They're talking about the protons flowing back down their concentration gradient to generate ATP. Only about 34%, and again, I'm not going to test you on exact amounts. It's more important that you understand that it's only about a third. It's a small amount. of the energy that's available in one molecule of glucose is going to be used for energy. And if you watch that Amoeba Sister video, one of the things it'll say is sometimes it says 32, sometimes it's 34, sometimes it's 38. It all depends on a lot of different conditions, the species you're talking about, if oxygen, there's a whole bunch of things that make this value 32 ATPs, sometimes make it 36, 30, it can vary. And every textbook I look in, just like the video said, seems to vary wildly. So you really just need to know that it's around 30 something molecules of ATP. And then the rest is lost as heat. So it's not a perfect system. And that heat gets lost, and we have to continually put more energy in the form of food back into our body so that we can get more ATP and also warm our body, because that heat, even though it's going to dissipate over time, it's going to be used to heat our body, to keep us warm, to allow all of our chemical reactions that are occurring with enzymes. It has to have a certain temperature, so we've got to maintain that body heat in order for that to happen. So everything has to happen with that heat. So here's the whole thing kind of shown in diagram. Glycolysis. This is a good overview, and now that we've gone through the whole process, should make more sense what's going on. We take glucose and we put it into, we make 2 pyruvates from it. We just get a little bit of ATP from that. Again, don't focus on the numbers. A little bit of ATP versus a lot. We're going to oxidize our pyruvate. I forgot to mention, we also have NADHs produced here. We haven't produced here. Never mind. I should clear the screen and do that again. We have some NADH produced there. We have NADH produced in oxidation of pyruvate. We have more NADH produced in citric acid. And then all of these guys are going to go. They're all going to oxidative phosphorylation in the electron transport chain to produce the bulk of our ATPs. And I see they say 26 or 28. It has to do with how many hydrogens are available, and that has to do with other things that we don't need to worry about. But, yeah, that's the summary. Many questions on aerobic with oxygen cell respiration. Fascinating, right? Oh, sorry, the door is locked. Iris, could you get that for me? I was trying to get rid of that beautiful music. Yeah, thanks, Ellen. Okay, so there's other kinds of cell respiration that do not require oxygen. And some of them are quite tasty, as it turns out. So we're going to talk a little bit about that. Most cell respiration depends on the electronegative pull of oxygen to pull electrons down the transport chain. Without oxygen, the whole electron transport chain is going to shut down, because then all these taxis, those shuttles, NADH and FADH2, they can't work anymore because they cannot become oxidized. Oxygen won't pull it away if there's no oxygen. If you're going so fast, if you're breathing if you're working so hard that now you cannot get enough oxygen into your lungs, the only thing that can happen is glycolysis. And for a short period of time, glycolysis can couple with, for us, it can couple with anaerobic respiration, or there are some critters who all they do is glycolysis and fermentation. Okay? And that's how they produce ATP. It's not the most efficient way to do it, but, in a pinch, it does work for us. And for other critters, it works for them always. In anaerobic respiration, there's a different final electron acceptor. And that could be, instead of oxygen, it could be sulfur, it could be other things. We call it anaerobic because you're not using oxygen. Yeah.
I have a question.
Yeah.
You
pass out, like when you like faint after like holding your breath? Is that because you're like?
Yeah, I mean, I've never like tried it on a kid who says I'm gonna hold my breath,
Yeah.
but yeah, it's part of our brain. When you pass out, you no longer have that physical control of holding your breath, and your body will just start breathing again on its own. Yeah, respiration will just start again. So you have anaerobic respiration, and you also have fermentation. And we're going to talk about each one of those. Fermentation, let's talk about first. And fermentation is where there's glycolysis. So glycolysis, again, I said last time, glycolysis is performed by every living thing on the face of the planet. It doesn't matter if you're a little microbe, a little E. coli, or if you're a little worm, or if you're a cockroach, or if you're a human being. We all perform glycolysis. But at the end of glycolysis, if you're not using oxygen, you have to figure out a way to regenerate your NADH back into NAD plus, right? Something has to take those electrons away because otherwise it's not going to be able to generate any more ATP. So there's two ways to do that, and those are listed here. The first one is alcohol fermentation. And in alcohol fermentation, yeast performs this. And yeast can be used for all sorts of things. One of the byproduct of alcohol fermentation is what? Ethanol, alcohol. Yeah, alcohol, right? They are not using that ethanol. That is their waste product. You can think about it that way. The yeast is producing a waste product, ethanol, that many people drink. Okay. Yeast also in their process. I'll just see if there's more slides on this, or if I need to talk about it. Okay, yeah. In lactic acid fermentation, pyruvate gets reduced by NADH to form NAD plus, and so that regenerates the NAD plus, so more ATP can get made. No, it didn't. Sorry, it didn't. It skipped over the ethanol one. Sorry, let's talk about alcohol for a minute. I thought that was alcohol. It was the other one. So the ethanol that's produced is used in wine, and it's used in brewing beer. And it's also used in baking because... Any bakers in here? Any bakers? All right, Evelyn. So if you want to make bread rise... Yeast. Because yeast is releasing... What? Carbon dioxide. So when those little yeasties fart, that makes your bread rise. Or burp. I don't know if they fart or burp, which one it is. And I've always heard you can either be a burper or a farter. Have you heard that? You're not both. You either predominantly burp or you predominantly fart. You can mull that one over in your head. I won't make you tell me which one you are. But yeast do release carbon dioxide as one of their byproducts, and that makes our bread rise. They are living creatures. You put them in with a little warm water. Evelyn, what else do you do with yeast when you put them to make bread? Yes, Evelyn. Yes. So if you put sugar with the yeast, that's their energy source. Just like we talked about glucose, right? That sucrose acts as an energy supply, along with some warm temperature, right? Because you can store yeast in the fridge or in the freezer, and they'll pretty much just go dormant. They're still active, but they're not doing too much. You give them some sugar, and they go, whoa, woohoo, here we go, and they start multiplying. They get a little froth on the top. You get that smell of nice yeasty smell for bread, that smell. And then you also get release of carbon dioxide, and that makes the bread rise. That is all. Also, sort again with the alcohol fermentation, brewing as well. Brewers have to be really careful when they're producing their alcohol, because their ethanol, because the yeast that they're using will give off gas. They'll give off carbon dioxide. So if you're trying to brew something in a closed container, why could that be an issue? It can explode. Yeah. So you've got to have some kind of release valve to allow that gas to dissipate and still keep everything else inside. Lactic acid fermentation. This is not done by yeast. This is done by us, as well as bacteria and fungi. At the end, we don't release carbon dioxide from this. We do make lactic acid. Lactic acid is partly responsible for the pain that we feel in our muscles, sort of. It's really micro tears, and a lot of people say, "Oh man, I got so much lactic acid in me from, lifting or running or whatever." It's actually tears. The lactic acid does a little bit of damage to your muscles and leaves little tears in your muscle, and then your body builds them up more strongly the next time. You go to work out, your body's going to be stronger than it was before because it's building it up more strongly than it had been in the previous version of itself. Lactic acid can also be used to make cheese and yogurt. If you look at your yogurt container on the side, it'll say something about contains active cultures. That means that there is bacteria in your yogurt. There's always bacteria in your yogurt. Okay. Same with cheese. Lactic acid fermentation is used to make soy sauce as well. Human muscle cells use lactic acid fermentation to generate ATP during strenuous exercise when you don't have enough oxygen. So when you start to feel that burn in your muscles, like I got to slow down here or I got to stop because I'm not getting enough oxygen to your tissue. That's the buildup of lactic acid. Once you stop whatever exercise you're doing or whatever activity you're doing, your body will actually take that lactic acid and put it back into cell respiration and take it through Krebs cycle and electron transport chain and chemiosmosis. Okay? But it won't do that until you stop so that you get enough oxygen back into your body to do that. Fermentation and aerobic and anaerobic respiration, all of these processes use glycolysis. Remember, we said that glycolysis gave us a whopping 2 ATPs. That's it. Wah, wah, wah. In all three, NAD plus is the agent that is becoming reduced. It's going to accept those electrons during glycolysis. The big difference between fermentation and cell respiration is the amount of energy that's produced. So we can't go very long performing lactic acid fermentation or going anaerobic, as we more commonly say, because we produce only, every glucose molecule produces, again, just 2 ATPs until we slow down. And then we can go back and we can perform cell respiration where we go all the way through all the steps and we get 32 molecules. So a lot versus a little. And we can only live, or we can only work with that little amount for a very... Is that why you can go without breathing? Yeah, so that's another thing. And you can train yourself. My athletes probably have to do some anaerobic training where you do intervals. Anybody do like sprint intervals where you go hard and you back off and then you go hard again, you back off again. What you're doing is you're training your body to go for longer and longer periods without that oxygen to tolerate more and more lactic acid. So that is a thing. There are some things we call obligate anaerobes. And that means they are obliged to, they have to perform fermentation or anaerobic respiration. And they either do very poorly or they just die in the presence of oxygen. If they have O2, they don't do so well. Okay. Yeast is an example of an obligate anaerobe. It does not like oxygen very much. It can live in oxygen environments. But when you're brewing, if you're brewing beer in particular, you want to, the brewers will very carefully monitor how much oxygen there is in the tank, because too much oxygen can kill off the yeast. They don't like too high levels of oxygen. They also don't like the ethanol. Yeast will be killed by ethanol because it's kind of like their waste product. So they're killed by the ethanol. Yeah, if you leave them in for too long. Many bacteria are what we call facultative anaerobes, meaning they can survive either by fermentation or cell respiration. We can survive for a brief period of time as going through fermentation, through lactic acid fermentation, but not very long. Well, how long can you go without? How long can you sprint? Pretty long? Wow.
Wow.
However long you can sprint, that's how long you can go anaerobic. That's how long you can tolerate your lactic acid fermentation. All right.
Wait, so lactic acid fermentation is a bad thing?
It's not a bad thing. It's our body's way of coping with the fact that we don't have enough oxygen.
Okay.
But our body does have to pay for it.
But isn't tolerated.
It's not very tolerated. Like I said, you can learn to tolerate more.
Yeah, you know, like those free divers that you hold for like
Mhm.
7 minutes. No.
Free diver. So here's a picture of showing. You start with glucose. Glucose is out here. This is our mitochondrial. That's our outer membrane. This is our. Oh wait, no, it's not. Sorry, this is our outer membrane. This is our mitochondria. Glucose comes into the cell. It goes through glycolysis. This is happening in the cytosol or the cytoplasm. It's becoming pyruvate. Pyruvate's the fork in the road. It's like, what do I do? If I don't have any oxygen, I'm going to go this way. If I have oxygen, I'm going to go the route of the mitochondria. Glycolysis is an ancient process. It says it right there, so it must be true. And we know that because if you look at what process every living thing, even the oldest critters that have been around forever, since the beginning of time on the earth anyway, performs glycolysis. Early prokaryotes likely used glycolysis to produce ATP because there was no oxygen. We do know that the early atmosphere did not have oxygen in it. And then somewhere along the way, cell respiration and fermentation became more prevalent as the oxygen started to build up in the atmosphere. Because now, if you have oxygen, now you can go through and do that whole oxidative phosphorylation. But we also know that couldn't have happened right away because you have to have what to do oxidative phosphorylation? Oxygen, and you also need the organelle. You need a mitochondria. And if you're a little bacteria, you don't have any mitochondria, so you can't do that. So very early organisms wouldn't have had the ability to do that. All right. I will stop there. We don't have too much left in this chapter, but I think we've done enough for today.